So in this, remember that normal blood sugar numbers, that 60 to 110 is kind of considered the norm. Diabetics can routinely run much higher blood sugar numbers. Hyperglycemia is really where you end up causing the problems. Hyperglycemia is when you're above 110, but they can have blood sugar numbers in the 200s, 300s, 400s. I had a student that said they had somebody came into the ER, it was 600 was their blood sugar value. So that blood, you've had what? 1,500? It was 1,100. So that blood is like normal, think of 100. Yours was 1,500 times or 15 times thicker than it should have been. So you think just how much problems that ends up causing because that thick blood doesn't flow, heart has to work harder, increased risks of stroke and heart attack under those conditions, and then you end up having chronic capillary damage. So the little delicate blood vessels end up blowing because this thick blood is flowing under such high pressure, and so that can end up causing blindness, causing kidney damage, leading to the neuropathy, all of those other conditions that patient is going to be dealing with, all because of poorly managed diabetes. And for the type 1 diabetic, it is really difficult to manage because your diet changes so much. You don't eat the exact same amount of the exact same thing every single day. So the type 2, though, is 90% of diabetics. So I got the numbers. So I was telling you how they can have periods of hyperglycemia. It is typically not as bad as what you see with type 1, but they can still end up with capillary damage, heart disease development, hyperglycemia leading to hypertension, kidney damage with that. The big sort of management options that they try to push is to really change the diet, to try and limit the simple carbohydrates, to limit what's going to get absorbed really quickly and what's going to cause dramatic effects on your blood sugar, and then also the act of trying to exercise to try and decrease body fat, because they have found that with type 2 diabetics, many of the times, about 80% of your type 2 diabetics, it's because receptor numbers decrease. So the insulin receptors right here, because of higher than normal body fat percentages, those receptors stop being made. So the cells literally take them off of the cell surface. And so I said you can think of insulin as being the key. The receptor is the lock. So it's like it takes the lock away. So therefore that body cell has not even got the option of getting glucose because there is no insulin receptors on the surface. And so the body shifts over to burning fat instead, and typically because of high body fat percentages. So one way that they monitor it now is they look at what's called A1C. So A1C is the average glucose concentration over 3 months. So it is actually the glucose in red blood cells. So they draw blood, they separate the red blood cells, they bust them, and they actually measure the glucose concentration inside of the red blood cells, because your red blood cells last about 120 days. The glucose level in the red blood cell is going to be consistent, so they can actually monitor, did you have times when you were making red blood cells when your blood sugar was really high? Because more sugar will be in that red blood cell under times of hyperglycemia. So it actually gives them a better picture of if they've been doing this or if they've been pretty steady all along. So numbers of 4 to 5.5 or 5.6% is considered normal. So most people have A1C numbers under 5.6, but from 5.7 to 6.4, they are now calling you a pre-diabetic. So that is a way of actually being able to identify somebody that has had periods of hyperglycemia over the last 3 months, which shows that either the receptors are not working as well, the pancreas is not secreting insulin, so it's helping to identify them earlier. Because if you can identify in that 5.7 to 6.4 range, then you can ward off some of the damages, the chronic damage that occurs because of hyperglycemia. So other ones that we talked about in monosaccharides. So glucose, most common sugar found in fruits and vegetables. It's one of the it is the sugar that is used to build all of the polysaccharides. But galactose is a sugar that's almost identical to glucose. It's really only different in the alcohol position. Galactose is found combined with glucose in milk. So galactose plus glucose makes lactose. This formation of lactose is a condensation reaction. So remember that water is removed to link these two single sugars together. And then we talked about problems that people have if they don't make the enzyme to break that disaccharide apart. So lactose intolerance can occur. Mannose we listed. Mannose is kind of unique and different. It is not a common monosaccharide, but it is a sugar that's found in cranberries. So it actually gets absorbed, it doesn't get utilized for energy, so instead it gets filtered out by the kidneys. So you will have mannose then getting excreted, but they found as mannose moves through the urinary tract, it actually helps to pull bacteria off the walls of the urinary tract. So somebody with a urinary tract infection, this can be a way of alleviating symptoms, alleviating irritation in the walls of the ureter and the bladder, which are all sort of common symptoms that people have when they have a UTI. And so that's where the old wives' tale of drinking cranberry juice when you have a UTI came from. And in fact, some people like take cranberry pills, so you actually are getting a concentrated source of mannose. You're just swallowing a pill with mannose in it instead of having to drink the cranberries. Some people don't like cranberry juice. But that's really like, when you look at it, that's, again, the only thing that makes this different from glucose is the position of that alcohol, that first one. Remember, glucose is right, left, right, right. That's the positions of the alcohol in the normal D-glucose. And so D-mannose is just one different, but again, not commonly found in all fruits. Fructose is the one that I told you is a ketose. So all these other ones have been the aldehydes. They have been aldoses. This is a 6-carbon ketone group, so they called it a ketohexose. Fructose is the one that they commonly call fruit sugar. Okay, so glucose, they usually refer to it as blood sugar. Fructose is considered fruit sugar. And in fact, fructose is the sweetest of all of the sugars. And so you detect sweet where? What part of your body detects sweet? The receptors. So the taste cells on the receptors of the taste buds are what detect it. So the shape of that ketohexose of fructose is detected about 1.5 times sweeter than table sugar. So it's really the sweetest natural sugar that is known. So you can use fructose for energy. So like I told you, galactose actually gets converted to glucose and then can be used for energy. Fructose can actually be used directly in energy production. And we'll talk a little more about that in chapter 12. The 5-carbon sugars, there's really just these. These are the more common ones. So there's ribose and deoxyribose. So these have this importance because they're pentoses, 5 carbons. They're both aldopentoses because they have an aldehyde group on the far end. And you find them as the part of the chains in nucleic acids. So the ribose is what you find in RNA, and that's the reason why RNA is ribonucleic acid. So the ribo part is because of that sugar, whereas DNA is deoxyribo, which is referring to the sugar that you see on the right. And the only difference between those two is that one's missing an oxygen. One has been deoxied, which is the reason for that name and the name difference. So if you look at the ribose and deoxyribose, you see it's exactly the same. Same number of carbons, same pattern. The only thing that's different is that second carbon in ribose is an alcohol, and in deoxyribose, that's a hydrogen. So it's just lost in oxygen. It's really the only difference between. So one characteristic that monosaccharides can do, these aldehydes, and ketones, they have this double bond oxygen. They call it the carbonyl is the group. So you can actually alter that end group if it's an aldehyde, and in fact the ketone can get affected as well. You can oxidize by adding oxygen. So if we oxidize, we take and add oxygen to the aldehyde and convert it to a carboxylic acid. So I told you this is what happened with the dipsticks in lab. So the glucose dipsticks that we used, if glucose was present in the solution, then the chemical would oxidize and change color. On the opposite side, you can remove hydrogen. And this one, adding hydrogen is reduction. I was thinking removing hydrogen is oxidation. Adding hydrogen is reduction, and it will actually change this oxygen, the double bonded oxygen, will get changed to an alcohol group. So they call these sugar alcohols. So sugar alcohols are alcohols like xylitol, sorbitol, erythritol. Those are all the sugar-free chemicals that you find in like sugarless gum. So they still have a sweet flavor, but they don't actually get broken down for energy. So that is why they can call it sugarless gum, and it has very low calorie content in it. So if you oxidize the sugar, you can make the carboxylic acid group by adding an oxygen to it. Good example of that is the dipstick test. But you can also reduce the sugar, and if you do that, then you add hydrogen, which converts the double bond oxygen to an alcohol group, adding hydrogen to it. So this was the example that they used. So we use the ones where we had a yellow to green color change, but this one you can see the same thing. Normal, the test solution they use is called Benedict's reagent, and it's got a copper solution. If there is an aldehyde or a ketone group that can react, it will convert that copper ion into a solid copper oxide, and it changes color. So very similar with the dipstick. You get that change of color. The more glucose is present, the more color change you'll end up with. So we're talking about disaccharides. So disaccharides are two. Disaccharides have two sugars linked together, but when you see them, they're not linked together like long chains, they're linked together as rings. So remember that when we drew them, like in the very beginning, like showing them, you had like this ring structure, and then you would see that it was connected to another one. Well, each of those is a sugar, but we've been talking about these like long straight chains of looking at fructose or looking at glucose, and so it all goes back to the shape. We'll start with D-glucose on this side. So this is the chain, 6 carbons in a row. The end one is that double bond oxygen. And remember I told you that double bonds are more reactive than single bonds? Double bonds are a little less stable. So this double bond is a little less stable than any other bond in the molecule. So this molecule could be more stable if that double bond could open up. The way it does that is to form this ring. And so in this, if you take and remember that these carbons have that tetrahedral, remember how the tetrahedral makes kind of a zigzag pattern? So if that carbon, the very first carbon, if it bends around, it actually lines up. So it's right next to the 5th carbon. So you'll have the first carbon, it'll line up so that the 5th carbon and the first carbon are actually really close together. In order to be more stable, this hydrogen that's on the alcohol group gets moved over to the double bond of the oxygen on carbon 1, and that's going to create more stability. By forming the ring, the oxygen on carbon 5, do you see how this one's blue? Just like this one is blue. So that oxygen on carbon 5 becomes part of the 6-membered ring. They always put that little oxygen in that upper right-hand corner. The way that you draw this always starts with that, whichever carbon has the double bond oxygen, it's always on the far right. So in this one, carbon 1 is always going to be on the far right. That's carbon 1. The way that the ring forms around, carbon 2 comes down, carbon 3 goes straight across, carbon 4 looks like it's straight across from carbon 1. Carbon 5 is the one with this alcohol group that's like sticking out over on this side. This is carbon 5. Now remember, carbon 6 is not chiral. Carbon 6 is, they typically just draw that it sticks up off of that upper left-hand side. So that's the 6th one. So when you go to draw it, always put your carbon, start off carbon 1 over on the far right corner, come down to the left, straight across to carbon 3, come up to the left to carbon 4, and then back around to carbon 5 to form the ring. This oxygen right here becomes the link. So the oxygen of carbon 5 forms this hexagon shape, kind of completes the hexagon shape. So when this happens, and sometimes it'll open back up, but it can close and it'll open back up. It's actually more stable as a ring, but the only difference is carbon 1 can have an alcohol group when it goes into its ring shape. The alcohol group can stick up or it can point down. So notice how this one sticks down. Notice how this one sticks up. And it can be either way, it just depends. So when this closes over, the alcohol group sticks down. Or when this closes over, the alcohol group sticks up. So it's either one. They're about even split. So they said, well, if it sticks down, that alcohol group is then considered the alpha form. If the alcohol group points up, then that's the beta form. So you see how they call it alpha D-glucose and beta D-glucose. That's really just what the alcohol position group on carbon 1 is. So we had the exact same. Carbon 1 is here. Come down to the left to carbon 2, straight across to carbon 3, up to the left to 4. So they just naturally take that tetrahedral kind of bend to it. Carbon 5, and then up 6 is just CH2OH. The oxygen of 5 is what forms the link. That oxygen of 5 is the link, and if we have a beta glucose, the alcohol group on that far right carbon is going to stick up. If it sticks down, it's alpha. So remember, beta is up and alpha is down. Beta is up and alpha is down. The position of that alcohol group is the only difference between those two. So the next ones here, figuring out where the hydrogens and the alcohols go on these other carbon groups. So carbon 1, if it points down, the alcohol group down would be alpha, alcohol group pointing up would be beta. But then carbon 2, 3, and 4. Remember that those are chiral carbons. Those are ones where they have 4 different groups, and so you can have mirror images. So they end up actually like being stuck in a conformation when you go into the ring position. Everything that's on the right-hand side of the straight column points down. So groups on the right with carbons 2, 3, and 4, they all point down. So carbon 2, the alcohol group, is sticking off to the right, and so it's going to point down. Point down. Hydrogen on carbon 3 points down. And the alcohol group on carbon 4 points down. Points down. So these, they're all on the right side of the chain. They're all going to stick down, always. Glucose will always have that position because those are the chiral carbons. So the ones that are over on the left, they all stick up. groups on the left of C2 to C4. These are going to point up when you make a ring. So the hydrogen sticks up. Jeez. Alcohol sticks up. Hydrogen sticks up. Same thing for the top one. Hydrogen up, alcohol up, hydrogen up. It just depends on where it ends up. So if you see the alcohols pointing down, that's alpha. If you see the alcohol pointing up, that's beta. And it's random. So if this ring opens up into a chain and closes up, it's random. Sometimes the alcohol group will end up down because it all depends on the position when that ring closes in.
So you just have to follow the ring and look at where their position to see whether or not it's
just for that first carbon. So it's alpha or beta is determined on that first carbon. Carbon 2, 3, 4, it's always exactly the same. So you see that on carbon 2, the alcohol always sticks down because it's on the right-hand side. Hydrogen's pointing down for carbon 3, alcohol group's pointing down for carbon 4. Because it's on the right side of the straight chain and they're all chiral carbons. So their position is like defined. The only one that can be iffy is the first carbon. It could be beta if it's sticking up, alpha if it's sticking down.
So if you're just looking at that, how would I know that that's an alpha? If I was just given that, how would I know?
If you see the alcohol pointing off from that far right carbon, that's alpha. So let's do this one. Okay, so here's another one. So first put down the 6 carbons. That's what I always do. And to me, it looks like an upside down question mark. So I go carbon, 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 and then CH2OH. Okay, so it's like an upside down question mark. And then number. This is 1, 2, 3, 4, 5, 6. Carbon 1, 2, 3, 4, 5, 6. Always start over on the right corner, then come down. So like I said, always like arrange it so that it looks like this upside down question mark. So in this one, you want to draw alpha D-glucose. So the next thing, well, I've got to connect it to make here's my oxygen. That's going to make the hexagon. So there's an oxygen as part of the hexagon ring. So it's really not a cyclohexane, because I've had some people say they thought it looked like a cyclohexane. Because it has the oxygen in it, it's really not. But I said if it's alpha, that tells you where carbon 1 alcohol is. So for alpha, the OH on carbon 1 points which way? So that means OH goes down. If the OH goes down, then what's sticking up? The hydrogen. Right? Because wherever there's always they've always got an H and an OH. When you form the ring, there's always the H and the OH. Okay? So now look at carbon 2. What's going to point down? So the OH is going to point down. What's going to point down in carbon 3?
H.
And then carbon 4? And you can write it HO or you could write it OH. It's the same. Sometimes you'll see it drawn this way because then it looks like the carbon connected to the oxygen connected to the hydrogen. Whereas if you have it drawn this way, do you see how it looks like the carbon's connected to the hydrogen connected to the oxygen? Like if you drew it that way, you really have to make sure that your carbon goes over to the O. Just because oxygen always has 2 covalent bonds, hydrogen never has more. So if you see it drawn like this, that's the same thing as an alcohol. It's just written backwards. Okay, in OH or in HO, same thing. But that's how I knew carbon 2, the alcohol points down, carbon 3, the hydrogen points down, carbon 4, the alcohol points down. That's how I know those, just from looking at the structure. And you'll always have that. In fact, really all you'll have to do is recognize. Okay, I'm not going to ask you to draw it, but I will say, what is the correct form of this sugar? And so you should be able to look at the straight chain sugar and look at the rings and be able to pick it out. So you always look at the middle carbons. Carbons 2, 3, and 4. The alcohol or hydrogen groups that are on the right will always point down, so you can pick them out. The alcohol or hydrogen groups on the left, these ones will always be the ones that stick up. Okay? So the only one that doesn't, which carbon doesn't have 4 single bonds? There's only one that we didn't add. You see it? All the carbons have 4 single bonds except carbon number 6 is CH2OH, so there's 2 hydrogens and an OH, so it does have its. No, one has a hydrogen and OH connect to an oxygen, connect to a carbon. So you see 4 lines with 2, 4 lines with 3, 4 lines with 4. Do you notice this one? See, there's only 3. This one, carbon 5. Carbon 5 is the one that its oxygen is over here. And so this hydrogen is going to be sticking off of carbon 5. So that one doesn't really fit the up and down, but that's because of the position of that big carbon group on 6. So that's this. So this is the single carbon off of carbon 5 is that hydrogen. So you just really need to be able to pick it out, okay? So in looking at them, we'll do a couple more next time to see. So here's fructose. So fructose is a little weird because fructose has that double bond oxygen in the middle. So this one, we want to draw beta. So in this, it is carbon 2 and carbon 5 that end up pairing. And instead of being a hexagon, this is going to look like a house. This is going to look like the pentagon shape. So carbon 2 is going to be the one that's over on that far right corner because it's the one with the double bond oxygen. Carbon 3 will come down, carbon 4 across, carbon 5 will be over here, and the oxygen of carbon 5 is the one that forms like the point, the top of the house. So this one to this one to this one. And it's because it is a ketose. So instead of making a 6-membered ring, it's still got 6 carbons, but it's going to make a 5-membered ring. So sticking off of carbon 5 will be CH2OH. Just like it was with glucose, the last one sticks up. If we want to do beta, remember that beta, the alcohol group will stick up. So if the alcohol group sticks up, then that tells you carbon 1 sticks down. And see, carbon 1 is also a CH2OH. It's not chiral, so it doesn't have to get drawn out differently. It can just get listed. So it's like a carbon group sticking up, a carbon group sticking down, and then has that 5-membered ring. But same thing as last time. What sticks down off of carbon 3? Just like before, H would sticks down. What sticks down off of carbon 4? OH. And you could put an HO or you could put OH. Doesn't matter. Just so long as the carbon's connected to an oxygen. It's just an oxygen hanging off. So what sticks up off of carbon 3? Over here. An OH or an HO. Doesn't matter. And then what sticks up off of carbon 4? H. And so here's carbon 5's. Let's see what color. So carbon 5, this is what sticks down. That's what hangs off. That's kind of like it's up in the top corner of the ring with glucose, but with this one it would stick down. So really, this is the only 5-membered ring one. It's fructose, because maltose, or not maltose, but galactose and glucose are really the other 2 that make the disaccharides. But if you see one that's a 5-membered ring, that's fructose. So when these, and there's examples of how they form. When we take two monosaccharides, they have to be in a ring in order to link together. And so the link is condensation. So we talked about that in chapter 5. So remember, that's when you have two molecules, two groups. They both need to have a hydrogen or an alcohol group available. So sticking off of those groups, you're going to have an oxygen and a hydrogen on either side. So in condensation, we're going to remove water. So out of that, you'll just pull two hydrogens and one of the oxygens off, and that forms the water, and that's going to leave one oxygen to become the link. So it's going to form like an ether connection. Linking those two together to form the disaccharide. So it's really the only reason to go through, like, well, how do they form this link? And this is why. That link is called a glycosidic bond. So think glycosidic and glucose. Like, so you think of the links between sugars. Okay, glycosidic bond is the links that hold sugars together into disaccharides and then polysaccharides. So here's the first one. So the first one is maltose. Maltose they call grain sugar. Maltose is made when two alpha D-glucose molecules link together. And so see the alpha? See the alpha. So remember I said alpha means the alcohol on that far right carbon sticks down. So when it forms a link, they call it an alpha glycosidic bond. And it is in fact linking on this glucose. Which carbon is the link formed by? Carbon number what?
1.
Carbon number 1, because remember it's 1, 2, 3, 4, 5, 6. So in that first one, it's carbon 1. What about the second one? Which carbon is this alcohol hanging off of? 4. So 1, 2, 3, 4, 5, 6. So they call this an alpha 1 to 4 glycosidic bond. So it means it's linking together in an alpha connection, the first carbon of one glucose and the fourth carbon of another glucose. Alpha glycosidic bonds will always have this kind of V shape. And it's because the alcohol groups pointed down. So they both pointed down, so they made this sort of V shape when you pulled the water off and made the link. So they call it an alpha link because this is the alpha position of the alcohol group. And it's a 1 to 4 connection because we're connecting carbon 1 to carbon 4. Carbon 1 of the left sugar is connected to carbon 4 of the right sugar in forming that connection. So this is how they defined different types of glycosidic bonds. So look at cell bios. So cell bios is made by some insects. How is that one different? So here, remember the alpha group is when the alcohol points down. So the alpha will always start off down, but that one is a beta. So this was a beta D-glucose. See, the alcohol group was pointing up when it formed the link. And so beta glycosidic bonds, they always kind of look diagonal. They always create this sort of more diagonal shape. So you will see them looking like that. Like linking two. So instead of them being side by side, they're kind of like this because they link up at a diagonal together. Whereas when you have the alpha link, it typically has like a V. And so they're side by side. So you notice with maltose, the two glucoses sit side by side and there's a little V connecting them. But with cell bios, they're a little bit, one's a little above the other one. And you see in diagonal type of link. If they're trying to get it on a page and not have it take up so much room, sometimes you'll see it drawn like this. Because then, see, they can still put the sugar. So they can still have this sugar and this sugar in a line, but you'll see this it looks almost like a zigzag or a Z kind of kind of shape, but it's still trying to depict the same thing. So remember that a beta 1 to 4 is going to have a diagonal appearance, and alpha 1 to 4 is going to have a V-shape appearance. That doesn't seem like a whole lot of difference. We make enzymes that digest alpha 1 to 4 linkages. We do not make enzymes to break the betas. So we can't break a beta 1 to 4, we can't digest it. So it's kind of like lactose. If you can't digest it, you don't absorb it. So if we eat cell bios, which like wouldn't be all that good anyways, because it's like some stuff you find in wood. But in that, we don't actually make the compounds we don't make an enzyme to break it. So maltose, they call malt sugar. It is typically formed when starches get broken down. So what they did is they would take a grain called barley. So they found with barley, they took barley, took the seeds, still had the covers on them, the whole 9 yards. So it's like you can see in that picture. So it really just looks like grain, like just harvested. It hasn't been peeled away. It's not just the flour component. It's the entire thing. So they would roast it. So they'd put it in an oven and heat it. Okay, and so you would like dry it out that way, and it would go from being kind of this pale tan, and it would get darker and darker and darker. So they would take and roast barley. Then they would soak that barley in water. When they soaked it in water, enzymes in the barley grain would begin to break the starch down within and would make maltose. And that maltose is what they would use to make beer. Okay, so that is like, so the amount of roastiness is actually how you get it different colors. If you've ever had a brown ale or a stout, that is where they've taken barley and they've roasted it at higher temperatures for longer periods of time, and it makes that grain really dark, and that dark color is actually given to the beer. But they call maltose then, sometimes some people call it beer sugar. I always think of like the Schlitz malt liquor because the malt in it like always helped me remember that it was this. It's 2 alpha glucoses, alpha D-glucoses linked together in an alpha 1 to 4 bond. And we do make an enzyme to be able to break this. So if you like put maltose in your mouth and you like chewed it up or like let it sit on your tongue, enzymes in your saliva would break it down and you would notice that it would taste sweet. It's not super sweet, it's not like fructose, but it still would taste sweet to you because you can actually digest it into glucose. So I always think of maltose as beer sugar. So lactose. Lactose is the other disaccharide. There's 3. We have maltose, lactose, and sucrose. Those are really the 3 most common. So look at the difference in this one. Lactose has a beta galactose. So notice that the bond sticks upward. Okay, remember I told you beta alcohols is when they point up, alpha alcohols point down. So this one, the beta alcohol sticks up and it's connected to a glucose. So it creates, again, the beta creates that diagonal sort of look to the molecule. It's a galactose linked to a glucose. The only thing that makes galactose different than glucose is this alcohol. So on glucose, the alcohol sticks down, but in galactose, the alcohol sticks up. And as long as you make amylase, as long as you make lactase, then you can break this. So like we talked about that, missing that enzyme in chapter 5, if you're lactose intolerant, you don't make the enzyme to break that beta 1 to 4 glycosidic bond. And if you don't break it, you can't absorb it. We can absorb the monosaccharides without any processing. Disaccharides have to get broken down into single monosaccharides to be absorbed. The last one. So the last one to me looks like 2 sugars stacked on top of each other. The top one is alpha D-glucose. Alpha, so see how its alcohol group was pointing down when the bond was formed. The alcohol was pointing down. And it's a D-fructose. So remember D, the alcohol group was pointing up. So like one's pointing down and one's pointing up. So they sit right on top of each other. in order to be linked together. So they look kind of like a stacked unit. The naming for them, because this is where the two carbons that used to be double bonded oxygens end up being linked, rather than just a carbon as another part of the ring. So they call this alpha because of the glucose, beta because of the fructose, 1 to 2. Carbon 1 is connected to carbon 2. So in this top one, carbon 1 with an alpha position is connected to carbon 2 of fructose, which is in the beta position. I always think of them as looking like they're stacked. Stacked sugars. Sucrose is the most abundant disaccharide found in nature. Sugarcane, sugar beets. So we grow large amounts of sugarcane. They take the interior, they grind it up, they dry it. It is table sugar. So when you buy a 4-pound bag of sugar, it is sucrose. It is a combination of glucose and fructose linked together as a disaccharide. And the link is a little different because it's both the carbons that used to be double bonded oxygens when they formed the ring. It's those two carbons that get connected. Again, you have to digest sucrose. So you make an enzyme called sucrase. And so it comes out of the pancreas. So this takes a little while. It's not, all it has to do is just clip that one glycosidic link. All we got to do is break glucose and fructose apart, and then you can absorb both of those. But it does require processing. All disaccharides have to be broken down. So the topic with disaccharides, really the big topic of the monosaccharides was diabetes. The topic of the disaccharides is sweetness. So the sweetness scale is all based on sucrose. So table sugar, sucrose, has a sweetness of 100. So that is like how your taste buds responds, the level of amount of sweetness that is described. Table sugar, they gave a reference value of 100. Now, sucrose is glucose and fructose linked together, has to be digested before it can be absorbed. Sucrose used to be the sweetener used in everything. So if you made cakes, if you bought a cake, it was made with sucrose. If you put any kind of sweetener in anything, it was always table sugar. Okay, like in all of your cooking, it would just be table sugar. But what they found was this. Fructose is about 1.5 times sweeter than table sugar. So that means you use less. You don't have to use as much fructose. And so for a baking company that was like making snacks, making cookies, making candies, things like that, using fructose, you don't need as much. So you could just add some other type of filler. You wouldn't have to use as much sugar if you use fructose because the person's tongue would think it was 1.5 times sweeter. So you could use less of it. The other thing that they found is that fructose, when you bake with fructose compared to sucrose, you get better browning, like it caramelizes. So baked things will caramelize. And so, you know, when you see something that's like got that golden color, you're like, oh, that's really pretty. Okay, so it's like got more physical appeal. So back in the 40s and 50s, like everybody cooked with sucrose. So then came along this understanding of changes began to occur, especially in pre-done snacks, treats, cakes, cookies, those types of things. And they began to use high fructose corn syrup. So that's actually, if you've ever used a bottle of Caro syrup, have you ever cooked, like if you make a pecan pie, you use Caro syrup. It's that super thick liquid. You can get light and dark, but it's a super thick sugar liquid, almost like honey. That's high fructose corn syrup. High fructose corn syrup is like digested sucrose because it's half glucose, half fructose. But the fructose in it is going to do what to taste? So if it was sucrose, if it was a disaccharide, it would have that base sweetness of 100. But if you break that and you actually use high fructose corn syrup, you're really just giving you already digested sucrose. So you're getting a mixture, half glucose, half fructose. And the fructose part is going to up the sweetness. This gets absorbed quickly because it's monosaccharides. They don't have to get processed. It doesn't have to get digested before absorption. In fact, glucose can get absorbed through the lining in your mouth. So if people, their blood sugar drops, what do you give them? You can give them candy and they can just suck on it. So it actually gets absorbed through the oral mucosa, through the lining in the mouth. It'll get absorbed rapidly from the stomach. Fructose takes a little longer. It doesn't get absorbed until you get into the small intestine. But if half of it's glucose, what's that going to do to your blood sugar? Spike it. But if you eat table sugar, that's got to get digested before it becomes glucose. So that will even give you a little bit of a delay. It won't give you a whole lot. You might have 45 minutes. When I was a kid, my parents didn't buy us any sugared cereal. We got cornflakes. We got unsweetened mini wheats, the plain ones. And we got Cheerios. Really, we got Tastios. Because my dad was if I buy the cereal they eat it all. So he would just buy the generic brand. Those were pretty much what we had in our cabinet. We had cornflakes, we had unsweetened mini wheats, and we had Cheerios. So my brothers and I have an older brother and a younger brother who taught me all kinds of tricks. So they were this is terrible. So what would we do? We would take tablespoons of sugar and just sprinkle it and then just pour a little bit of milk to wet it all down. So we were drinking this sugared milk, but it was sucrose. So we were fine on the bus. But by the time we got to school, then that sucrose was getting digested. And so you would get to school, you'd be ah, you're super hyper. But how are cereals different now? What do they use? They don't use sucrose. They use high fructose corn syrup. And so the kid literally is hyper before he gets off the table. Okay? When he gets up to go get his books and stuff, he's already okay? Because it's high fructose corn syrup, it's already monosaccharide size. And so it's all ready to get absorbed. So you're going to start absorbing the glucose in that high fructose corn syrup in the lining of the mouth. And so you'll see the blood sugar spikes immediately. So you don't have to wait. So it wasn't a bad thing for my mother, probably, because we were hyper by the time we got to school. But kids now, they're hyper before they even walk out the door because of that. The other thing, they've done a lot of research. High fructose corn syrup also triggers pleasure sensors in the brain that also sets up behavior. And this is why, especially if you're one of those people that has the behavioral tendencies, you have ice cream one night, you have ice cream the same time the next night. Third night, what are you thinking? It's time for some ice cream. It just becomes this habit, and it's the sugar. So the sugar, the brain is this is wonderful. So the brain says, this is delicious. And so it will actually begin to start and trigger behaviors. Really, you're just pleasure seeking. So you're seeking things that are positive, things that are good, things that are not negative. And so that becomes the sugar addiction. Because when you eat a lot of sugar, what happens to your blood sugar? When you eat a lot of this high fructose corn syrup stuff, especially, your blood sugar does what? Yeah, because half of it's glucose. So it's you're getting injections of glucose. And then your pancreas goes, oh my God, this blood sugar is 150. So then your pancreas does what? dumps insulin, so your blood sugar went like this, then what does your blood sugar do?
Correct.
Yeah, and it'll actually drop lower than it was before. So then how do you feel?
Correct.
Yes, then you get tired, so what do you go get?
Energy drink.
More sugar. So then you so it sets up this like cycle. So they have like been doing lots of research in the last 15 years. High fructose corn syrup has really like added to our sugar addiction, even more so than table sugar, because it's immediate. You get like this immediate blood sugar change from it because it's really like digested sucrose.
Is that why they say it's so bad for you?
Yeah. Yeah, because when your blood sugar goes up real high here, you got the hyperglycemia issue. But then your liver will also take that sugar and convert it to fat. So they find that people that are eating a lot of sugar have high triglyceride levels, that have a lot of circulating fats, and all that then all throws right into adding to the issues with heart disease, right? Having increased blood sugar is going to thicken the blood and put more pressure on the heart, more pressure on the vessels, and then also increase in triglycerides circulating. All that builds up the plaque, start to create the goop in those tubes, and that ends up leading to problems down the road. So yeah, they have been saying that these simple sugars, small amounts, not every single night necessarily, even though that's really hard to break. Because some people are like trying to break the sugar cycle. That's the hardest part, because you get on that train and you're like, but I need this, you know.
It's like a drug.
Huh?
It's like a drug.
Well, and it's your brain. Your brain like says, this is delicious. Like, so your brain then says, we want some more of that, you know, and it's right there. It's not like we're Little House on the Prairie people, you know? Like it's in the refrigerator, it's in the freezer. Like it's not difficult to stock up on such things, but you run into this long-term problem. And so you end up with like irregular energy levels and everything else. So looking down the rest of the list, that's kind of like the interesting disaccharide thing. Looking down the rest of the list, notice that glucose is actually not as sweet as table sugar. So blood sugar, if you taste glucose, it's kind of moderate, about 75 compared to sucrose. Going way down, maltose, galactose, lactose. So milk isn't really sweet, and it's because lactose has a very low sweetness scale. So your taste buds can tell the difference between maltose, lactose, and sucrose. 3 disaccharides, it can tell the difference because of the type of monosaccharides that make it up and the kind of bond that it has. Look at this craziness down here. So sucrose has a sweetness of 100. Splenda has a sweetness of 60,000. So that's why, like when you open a Splenda packet and you pour it out, how much comes out? Like 10 little powdery grains. It's not very much. There's never very much in those because you don't need very much. Your taste cells, they respond extremely strong to these.
Why is it supposedly so better for you?
Well, okay, so like there's some people that are sort of like, why would you want to eat a chlorinated disaccharide? So that's like, so Splenda is like sucrose with chlorine added into it. So you don't break it down, and if you don't break it down, you don't absorb it. So it's kind of like lactose. Some people still get some GI issues by eating or any of these artificial ones, because if your GI got bacteria like it and they'll ferment it, then they can cause similar negative side effects like lactose intolerant. Saccharin, now I will say like saccharin, they fed a rat like the equivalent of like 7 pounds of saccharin and it caused cancer. Like, I mean, it wasn't, they didn't, because they did all these animal studies. And so when they came up and they said saccharin could cause cancer, the amounts that they fed those animals was huge compared to their relative size. So it would be like you eating 7 pounds of saccharin a day. Like you eat 15 grams, like a teaspoon of it. That's like the norm. So that's pretty high, but it's another one. And here's the interesting thing. They were doing research on clothing dye, and someone tasted the dye and said, hey man, this is sweet. So they actually isolated saccharin from that. So it was like a fluke. It wasn't something that they weren't like doing research trying to find sweet tasting stuff. Someone was like, that's not bad. And that's how saccharin, because saccharin was one of the earliest ones. That's the sweet and low. So the pink, that was like Tab way back in the day. Huh? Do you remember that? Stevia is the only one in the list that's natural. So stevia actually comes from a plant. So when they first extracted it, it didn't go over very well because they extracted the whole thing and stevia was green. And people were like, I'm not putting anything green in my coffee. So they actually had to take it off the market. They had to extract all of the green chlorophyll from the plant. So they whitened it and then they made it nice and granular so it looks like sugar. So then people were like, well, okay, I'll try it. But initially it had a real light green tint because it comes from a plant. So they would just grind up the whole plant. The parts of the plant, they would just grind it. I think it comes from the leaves, but they would just grind up the leaves and the leaves are green. So then the stevia was green to begin with. They had to like take it off the market, retool it, extract the green stuff out because nobody wanted any chlorophyll in their coffee, and then repurpose it. And it's actually done very well, but you don't find it very much in like your sodas. You know, it doesn't do well with the carbonation. So you usually find like Splenda or saccharin or NutraSweet. So NutraSweet is one where it's actually 2 amino acids. So that's another one it's not naturally found, but amino acids are the building blocks of proteins. But they found these 2 amino acids together, aspartic acid and phenylalanine, as just a 2 amino acid group. Your taste cells respond as if they're sweet. The problem comes in, when have we talked about phenylalanine? Talked about it in chapter 5. Do you remember PKU? So remember we were talking about that enzyme that breaks down an amino acid called phenylalanine, and babies that don't make that enzyme begin to show neurological deficits. It causes permanent retardation. So anybody that is diagnosed with PKU has got to stay away from NutraSweet because NutraSweet contains phenylalanine. So if you're drinking a diet soda that has it, that's going to increase your phenylalanine intake.
So it is claimed that it can cause brain damage, right?
Well, but that's for patients that don't make the enzyme to break that down. So that is a very small percentage. It's like a 0.0001 kind of percent. It's not very many. And you get tested for it at birth. When they do the heel stick, they draw blood and they actually check to see if you have that enzyme. And if you don't, then they automatically put you on a diet low in phenylalanine and you don't end up with any of those symptoms. So that's really the biggest difference. So those last 2 are pretty sweet. The other 2, the Splenda and the saccharin, meh. But they are it just really depends on how they affect your taste cells. So you tell me, what is this type of linkage? So this is a beta. No, that's not a beta. Sticking down. Sticking down is alpha. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So this is alpha, right? So the alcohol was pointing down when it went through and connected. So you always look at the left sugar, look at whether or not it's pointing down or pointing up, okay? When you look at the glycosidic bond, look at the left sugar first. If you see that the right corner, look at the right corner carbon. If it is pointing down, then that's alpha. If it's pointing up, that's beta. Okay? So that's how you can determine what type of glycosidic link that is. So looking at this second one, this is carbon 1, 2, 3, 4, 5, 6. So this would be considered an alpha what? So carbon 1 is connected to carbon 6. Can you see that? Carbon 1 of the left sugar is connected to carbon 6 of the right. What about that one? Okay, so notice how this goes up. So that is a beta. This is carbon 1, 2, 3, 4, 5, 6. Same. 1, 2, 3, 4, 5, 6. So it's saying that it's a beta link because the carbon on the sugar on the left is pointing up as the glycosidic bond. So this one is sticking upward. That means that it's beta. This one is pointing down. That's the alpha one. It's connecting carbon 1 to carbon 4. So they call that a beta 1 to 4 glycosidic bond. What about that one? Alpha, because this one points down, so you know this is alpha. And it's a 1 to 4. Because this is carbon 1, 2, 3, 4, 5, 6. You always start and do your numbering, like always start on that far left or the far right corner and start the numbering that way. 1, 2, 3, 4, 6. So this one would be an alpha 1 to 4. Carbon 1 of one sugar is connected to carbon 4 of the other sugar, and it's an alpha position. So we'll do another one's the polysaccharides. So the last of this group, these are the huge ones. They sometimes are like, they call them storage. These are huge polymers of glucose. Polymers are lots of glucose units. They're typically alpha D-glucose, the ones that you find in starch anyway. So this first one, this is a plant. Plant form is starch. So plants put starch in the seed because that will be the energy for the new baby plant. So when you have, so if you look at a seed, there's a little tiny nub, like in a peanut. Have you ever the peanut has like that funny little single piece, and sometimes you crack a peanut and it falls out? That's actually the embryo, that little nub. All the rest of that is the food that little embryo plant is going to need. So what seeds do is they pack almost all of the inside of the seed is starch, and there'll be this little tiny embryo that's just a little nub in there. When that embryo germinates or begins to grow, it's going to use that starch for energy. So really, this is just a way to store glucose for the new plant. So this is a glucose storage molecule. Starch can also be called flour. Okay, so when you think of starch, think of flour. So when they take flour, they take all the covers off, so it has what's called the bran. So it takes the cover off and they grind it down. Now they usually, if you get white flour, they also bleach it. They bleach it because it makes your breads nice and white. So they would bleach it rather than use it just as is, because if you use it as it is, it's a little off-white, almost brown-ish looking, even if you take all of the covers off. So unbleached, it kind of is more like an ivory colored, the flour is. Okay? If you've ever bought unbleached flour, it's not brown. It's not like the whole grain flour, but it's not white, which is why they started bleaching it. But it's made of two different polysaccharide groups. The first one is called amylose. Amylose is only about 20%. Amylose is alpha 1 to 4 connections of glucose units in hundreds to thousands of glucose units in the chain. So it's a glucose unit, so one glucose unit with an alpha 1 to 4 connection to another one. So you just do that 4,000 times. So it's a really long chain of glucoses, and they're all alpha. So they all have that kind of V shape in their connection. This coils. So you just end up with this really long chain of glucoses. Now the other one is actually the more common type of starch polysaccharide. Amylopectin is one long chain, but then it has glucose units branching off the main chain. So you'll have the long chain, but then you'll actually have alpha 1 to 6 connections that form the branches. So here's an example. So the top one, that's amylose, and that's a really small amylose. So each of those little orangey colored blocks would be a glucose. Every single one of them is an alpha 1 to 4 connection. So they would all have that shape. So the connection. And the seed, when the plant builds Amylose, it just takes a glucose that it made by doing photosynthesis. It takes that glucose and just links it on, and then adds another, and adds another, and adds another. And it just keeps doing this till it gets this really long chain. And that's what it packs into the seed. Amylopectin has chains, but they branch. So still, the chains are alpha 1 to 4. But when you get to the branch points are alpha 1 to 6. And so remember, in alpha 1 to 6, like this CH2OH up here, that's that 6 carbon. So what it'll do is it will connect to an alpha of another glucose. So that would be an alpha 1 to 6 branch. So it's where the first carbon number 1 on the glucose connects to a carbon number 6 along the chain, and that's where you get these branches. So amylopectin starts looking very sort of big and more bulky compared to amylose. Good thing about this, we can digest all of those bonds. We can digest alpha 1 to 4, we can digest alpha 1 to 6. So we can break this. But this takes time. This starts off in your mouth. Saliva contains an enzyme called amylase. And amylase breaks alpha 1 to 4 links. So you put food in your mouth, and so any starch that's in there, you start to have the breaking. And so the breaking is, we clip them off. So it just begins to clip the alpha 1 to 4 links. Same thing in the amylopectin. It just begins to clip the ends, clips them shorter, usually clips them into maltoses, like clips them into 2 carbon group or 2 sugar groups. And so like if you take a plain cracker or a plain piece of bread, put it in your mouth, don't swallow it, just chew it and hold it in your mouth. It gets kind of gooey, but it'll start getting sweeter because the starch is not sweet. Starch is humongous. Flour, if you've ever put flour on your tongue, it's all pasty. It's because it's a huge molecule, so your taste cells can't get it. But once you start breaking it down, then you start to actually having maltoses, and maltoses will be a little sweet. So you'll begin to notice that sweetness of the bread. It does take a while though, so you can imagine clip, 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 clip. So it just keeps clipping them and clipping them. It doesn't happen instantly. In fact, this is why it typically takes hours. When you eat starchy foods, you're looking at digestion and processing of 2 to 4 hours for enzymes to be able to break these huge polysaccharides down to single sugar size. You can't absorb it until it's all the way to glucose. That means you got to break every single glycosidic bond. That takes a while. So that is why like having like more of the pastas not going to affect your blood sugar as fast. Okay, not as fast as the sweets, as the sugars, as those simple sugars. Those are going to get absorbed much faster. Here's another picture, and you can actually kind of see the chain in this one. So in that, the top one is amylose. Can you see how it looks like it sort of like curls? Like it makes like a coil? And then the bottom one is amylopectin. So you can kind of see that there's like a main chain in this one that kind of does the loop like the amylose does. But then you can see that there's these branch groups sticking off. So amylopectin is the more common one. Down here, it shows the links. Alpha 1 to 4, that's the V-shaped connections. All those glucoses are connected. But then the branches are this alpha 1 to 6 group. But you can digest all of this. It just takes time. So this is why a lot of people, like, if they're going to do some major type of exercise, they'll eat like pasta the night before. So like if you were going to have if you were going to run, if you were going to exercise, then you would like carb load, but you wouldn't eat like candy and cookies. You would eat starchy foods. So you'd have like a pasta meal because it's going to take a while for it to process, but it's also going to help you to have glucose for energy. That's really the big function of glucose in the blood and in cells. A storage polysaccharide that animals make, plants make starch, we make glycogen. And we don't make glycogen to pack around embryos. That's not necessary. We have like the whole umbilical cord for all of that mess. I mean, we do have a little yolk sac, but other than that, it's nowhere near like what a seed does. Because, you know, like we don't drop our fertilized embryos on the ground and hope they germinate. That would be easy though. But instead, for us to store sugar for the in-between meal times, we make glycogen. And honestly, glycogen looks just like amylopectin. So glycogen has alpha 1 to 4 links and alpha 1 to 6 branches off of it. So it really looks very similar. It's got a little more branching. So the branching is like every 12, whereas with amylose it's like every 25. So it's more branched even. Why you do this is for this. Glycogen can help to keep your blood sugar from dropping too low in between meals. So for some of you, like you might eat after this class, that might be dinner, okay? But then you get up in the morning and you start going. So you may not eat until noon just because of the schedule that you've got. So that means that we're talking like from 7 p.m. until 12 the next day. So you're looking at what, 17 hours, 16 hours in between meals? That is a long time. Blood sugar is always getting pulled out of the blood for use by the brain. Your brain doesn't need insulin to get glucose. So if nothing else, even if you're sleeping, you are still going to be using glucose for brain processing. So your blood sugar would start to drop. If you've ever had low blood sugar, you start getting kind of like sort of shaky and you're like, what? You're like trying to think about things and your brain's like, I can't think right now. There's no sugar. I have no energy. So this is where your blood sugar reserve is kept. When you eat carbohydrates, not only does insulin tell your body cells to take in sugar, it also tells your liver and skeletal muscles store excess as glycogen. So glycogen looks kind of like this. So see the one on the far left? That's glycogen. When you eat, glycogen gets bigger. So blood sugar will be taken out of the blood in your liver cells and skeletal muscles. You can make more branches. You can make this, you can just add on to the end, but it's going to get larger. It'll build, and then in between meals, you can break it down. So in between meals, your liver and skeletal muscles will actually clip off glucose and dump it back in the blood. Well, usually your skeletal muscles use it for energy, but your liver can actually help to make sure that your blood sugar doesn't go below 60. So when you eat, you increase glycogen size by adding more glucose. And then in between meals, you'll break that glycogen down. It'll get smaller and smaller, but that's going to keep your blood sugar up. So in between meals, you break glycogen down to try and replenish those blood sugar stores. So amylose is just a chain. So this is starch. So amylose is just that long chain. Amylopectin is the long chain with some branching. The glycogen's a long chain with lots of branching, like twice as much branching as what you see with the plant ones. And with this, the predominant place that you find it in the animals is in the liver. So your liver stores glycogen because it helps to balance out blood sugar in between meals. Helps to bring it down if it's high, but it also helps to replace it if it's low. So that way you can go 15 hours and not end up with low hypoglycemia, with low blood sugar issues because of this. All right, so I'm almost done. Hooray! So on Wednesday, we will finish chapter 6, and we'll talk about the first part of chapter 7. There won't be any math in it, it's just sort of a theory thing, and that'll finish up material for the first exam. I've only got like one last set, so we got to talk about cellulose and fiber. And then we're going to talk about blood typing, and we're done with chapter 6. Okay? So pretty straightforward. So go ahead and work on those. You should Finish chapter 5, finish the dynamic study modules. There's one on chemical reactions, one on redox reactions, as well as the post-lecture homework. And you can go ahead and work on chapter 6. There's the dynamic study module and the post-lecture homework, because we're almost done the polysaccharides. We're just about to the end of that one too.